Welcome to Biochemistry Simplified, where science becomes simple and engaging. Today, we're entering the world of biostatistics, a powerful tool used to interpret biological data. Whether you're analyzing experiments, clinical trials, or surveys, this video will equip you with the essential concepts and real-life applications you need. So stay tuned, and don't forget to like, subscribe, and turn on notifications for more academic clarity.
Hello, welcome to Biochemistry Simplified. We'll begin yet another exciting topic. I started deviated quickly into biostatistics for biological science due to curiosity and questions. So before I continue my biochemistry series, I said let me do this for those graduates and undergraduate students asking questions. And we already started it with the introduction for last week. Now we are continuing today on or to continue on sampling methods. But before I get into the real details of biostatistics, I want to look at what we call bioassay. So bioassay is what we'll be looking at today. Bioassay. So simply, what is bioassay? Bioassays are methods for the estimation of nature, constitution, or potency of a material by means of a reaction that follows its application to living matter. Okay, so it is basically defined as the estimation or determination of concentration or potency of physical, chemical, or biological agents by means of measuring and comparing the magnitude of the response of a living organism. You like to take that down. So we say bioassay. Bioassay is defined as estimation or determination is defined as estimation or determination so estimation or determination of concentration or potency of of physical, chemical, or biological agents by means of measuring by means of measuring and comparing by means of measuring and comparing the magnitude of the response, means of measuring and comparing the magnitude of the response of a living organism. Yes, as a detailed definition of bioassay. It is the estimation, the determination of concentration or the potency of physical, chemical, or biological agents by measuring or comparing magnitude of the response of a living organism. So bioassay or biological assay as it may or are methods used for estimation of the potency of substances by observing their effects on living organisms. So when their effects is observed on living organism, it is called an in vivo research or in vivo assay, or on isolated tissues. When it is on isolated tissues, it is referred to as an in vitro assay. And by comparing this effect of these substances of unknown potency to that of standards. So, bioassay can be said to be based on the use of biological detection system for biological active substance. So, like a quick um, shrinking or summary of this definition is that bioassay can be said to be based upon the use can be said to be based upon the use of biological can be said to be based upon the use of biological biologically active biologically active substances. Pretty short, right? So with all that is said, this is like a summary of what a bioassay is. So in its simplest form, antidote concentration of particular substance by comparing with a known amount of the same substance. All right. So both are procedures by which the potency of the nature of a substance is estimated by studying its effect on living matter. Bioassay is a procedure that for the determination of the concentration of a particular constituent of a mixture. So we quickly now look at what the components of bioassay are. Like we have said earlier, bioassay can also be divided into two components, which is, I'll do that on the next slide. So we look at the components of bioassay and we can say that the bioassay can be said to be of two types, which is qualitative assays and quantitative assays, like we saw in our last class. So biological sciences, like I told you that. So bioassay could be qualitative. The assays could be qualitative assays, and it also could be what? Quantitative assays. Okay, so we wrote like a small table here. So if we say it's qualitative, simply put, what it means is that these assays do not present any form of statistical analysis. So right under this qualitative assay, we see, because we have treated this in detail, so we just do this. Say these assays do not present these assays do not present any statistical analysis. They not present any statistical analysis. Sorry about that. Do not present any statistical analysis. All right. Why for quantitative assay? They provide numerical assessment, like we saw in our last class. We said statistical assays are numerically inclined. Statistical assays are numerically inclined. Statistical assays are numerically inclined. They provide numerical assessments of some property of the material to be assayed and pose statistical problem. So For quantitative assay, we write these assays. This provides first numerical. This provides numerical assessments of some property. Provides numerical assessments of some property of the material. Property of the material to be assayed. Property of the material to be assayed. And pose statistical problem. And pose statistical problem. Okay, so statistics, bioassay, like we said, can be quantified into these two groups, the qualitative and the quantitative assay. The active principle to be assayed should show some of the characteristics generally of bioassay is that the active principle to be assayed should show the same measured response in all biological species. Bioassay involves the comparison of the main pharmacological response of the unknown preparation with the standards. That's another characteristic. Another one is that the method selected should be reliable, sensitive, and reproducible, and should minimize error due to biological variation and methodology. That's another characteristics. So looking briefly at the characteristics of bioassay. Then the degree of biological response produced should be reproducible under identical conditions. Then we have the reference standard and test samples should have the same biological effects and mode of actions. So that those response curves run parallel and their potency ratio can be calculated. So I would like to quickly pen down some of these characteristics, if not all. So right here, characteristics of bioassay. Characteristics of bioassay. And we say number one is that active principles to be assayed should show the same one active principle to be assayed should show the same measured response in all biological species. So should show the same measured response in all biological species. So another characteristic we saw is that the bioassay should involve the comparison, or the method selected for bioassay should be reliable. The method selected for bioassay should be reliable. Okay, should be reliable. It should be sensitive. And importantly, and it should be what? Reproducible. It should be reproducible, and also should minimize error, and also should and also should minimize error due to biological variations. Should minimize error due to biological variations. Should minimize error due to biological variations and methodology. Okay, so we'll continue with the characteristics on the next page. Still on the characteristics of bioassay. Still on the characteristics of bioassay. And point number 3. So, point number 3, we say the reference standard and test sample should have the same biological effect and mode of action. Very key. The reference standard the reference standard and test sample should have the same biological effects, should have the same biological effects, should have the same biological effects and mode of action and mode of action. Then the fourth point we want to say is that activity assayed should be the activity of interest. Individual variations must be minimized and accounted for. Activity assayed should be activity of interest. Point number 4. Activity assayed should be the activity of interest. Should be the activity of interest. All right. What that means is that individual variations must be minimized and accounted for. Individual variations must be minimized and accounted for slash accounted for. Okay, so that's basically some of the characteristics. We still have some more. Bioassay might measure a different aspect of the same substance compared to chemical assay. That is one characteristic. The test solution and standard should be compared for their established biological effects using special biological techniques. Okay, so let me just give that as our last characteristics we want to look at. We said that bioassay might measure a different aspect of a substance of the same substance, of the same substance compared to chemical assay. Okay, of the same substance compared to chemical assay. Okay, very important. So it's not compulsory that chemical assay could give you a different result. So you might run the same experiments using a chemical assay, and it's going to be totally different when using bioassay, because it will be measuring a different aspect of the same substance when you compare it to chemical assay. They are more, like I said, the test solution and standard should be compared for their established biological effects using a special biological technique. That's another characteristics. Okay. Then, prior to the development of sensitive, precise, accurate chemical and binding displacement assays for the presence of concentration of drugs or octacoids, we had to rely on what? Bioassay. All right. So we quickly now move to the next um, segments that we want to look at, and this segment we'll be looking at the types of bioassay. I want to take another color of pen to do this. The type of bioassay. So types of bioassay. We want to quickly look at the types of bioassay. What are the types? There are mainly three types of bioassay other than the qualitative assay. The three main types of bioassay. So there are three main types. There are three main types of what? Bioassays. Other than the qualitative assays. That means qualitative assays is not included. So what are the three main types of assays? They include: number one, you have direct assay. Direct assay. That's a type of what? Bioassay. Number two, you have indirect assay. Indirect assay. And that's also another type of bioassay. And actually, this bioassay is actually based on what? Quantitative responses. This is based on quantitative responses. All right. And number three, which is the last type, is also a indirect assay, but this time it is based upon quanta response, all or none. Okay, so we have indirect assay, still on the type of bioassay, and this one is based on what? Quanta responses. Now, so what is direct assay? Direct assay deals with the doses of the standards and test preparation that are sufficient to produce a specified response and can be directly measured. The key word there is it can be directly measured. Okay, so it refers to the doses. So we quickly just take them one after the other. So in a direct assay, which is your number one, we want to look at. So we say that direct assay refers to the doses, of the standard, and test preparation, preparation, doses of the standard, and test preparation that are sufficient, that are sufficient, that are sufficient to produce, that are sufficient to produce a specified, that are sufficient to produce a specified response, a specified response that can be measured, that can be directly measured, and can be directly measured. And I said the key word here is directly. It can be directly measured. So in our next page, we'll look at indirect method based on quantitative response and indirect method based on quanta response. So we are right here. So we are looking at the indirect assay as a type of bioassay. Indirect assay. And we say that this assay is based on quantitative response. It's based on quantitative response. All right, so indirect assay, I would say, is based on quantitative response. So what is it? In indirect bioassays, the relationship between the dose and the response of each preparation is first ascertained. So then after that, the dose corresponding to a given response is obtained from a relation of each preparation separately. So indirect assay deals with the relationship. It is the relationship between the dose, and response. Between the dose and response of each preparation. Of each preparation. Of each preparation, and that this relationship between these dose and response of each preparation, we first have to be ascertained. So it's first ascertained or is first determined. Okay, it's first ascertained or determined. All right, then after doing that determination, then the dose corresponding to a given response, the dose corresponding to a given response, is obtained from relationship of each preparation separately. Is obtained from relation of each preparation separately. So that's basically what the indirect response is about. Then we also have the quanta indirect assay based on quanta response. And in that, we say that this response is in the form of all or none, means no response, or maximum response. So we are looking at indirect response based on quanta assay that can apply to either no response or maximum response. This can be bioassay by endpoint method. Predetermined response is measured which is produced by threshold effect. Quantile responses are population response based on all or nothing, that's 0 or 1, present or absent, response such as death. So concentration of unknown standard for quantile response will be now be equal to the dose of the standard over the dose of the test times um, on the concentration of the standard. So the next one, which is the indirect assay, still the indirect assay, and we say that it is based on quantile response, which is either response for maximum or nothing, so all or none. So this assay is typified by a relationship, and that relationship is seen as, it can be measured with this relationship known as the concentration, of unknown is equal to the of standard dose of standard over the dose of the test times concentration of standard times concentration of standard. Okay, concentration of standard over 1. So that's basically the relationship, and we say that that quantile response is the response that is in the form of all. So it's either you have no response at all in this type of assay, or you have maximum response. And we say it is bioassay by endpoint method predetermined by the response endpoint method, which is predetermined response that is measured and produced by threshold effects. So quantile responses generally are population response based on an all or none response, where all could be typified 1 or 0 or present or absent, and is such example is response such as death. So that's briefly on bioassay that we want to take on this lecture. There are much more details on bioassay, but quickly in our next slide, we want to look at what are the advantages and the disadvantages of bioassay. Okay, we don't want to go into much details, just giving you the basis. So what are the advantages and disadvantages of bioassay? Next slide. So we start with the advantages of bioassay. So advantages of bioassay. That's where we are beginning from right now. The advantages of bioassay. All right, so the advantages of bioassay, basically, you say it's sensitive of the tissue. Sensitivity of the tissue is determined by plotting, all right, of plotting a concentration response curve with known against. They not only help to determine the concentration, but also the potency of the sample. So one of the advantages is that in bioassay, you can determine the sensitivity of the tissue or sample you are researching on by plotting a concentration response curve with known against unknown. So they not only help to determine the concentration, but also the potency of what the sample. Advantages, we said that the tissue or sample being determined, it helps to determine the sensitivity of such tissues by plotting a concentration of response curve with known against unknown. They don't only help to determine the concentration, but they also help to determine the um, uh, potency of the sample. So we could write number one, so we say bioassay. Say bioassay helps in determining, helps in determining or in the determination, okay, in determining the sensitivity, helps in determining the sensitivity of the tissue, helps in determining the sensitivity of the tissue by plotting a concentration response curve, by plotting a concentration response, by plotting a concentration response curve, a concentration response curve with known, with the known with the known against unknown, with the known against unknown, okay. And this helps, this only not only help in determining the concentration, but helps, so with this type of assay not only helps, this helps, this helps in determining the potency of the sample. This also helps, this also helps in determining the potency of the sample. So it does not only help in determining the concentration of the sample, it also helps in determining the potency of the sample. So another advantage of bioassay, number two, is that it is specially used to standardize drugs, vaccines, toxins, or poisons, disinfections, antiseptics, etc., as these are all used over biological system in some or other form. So it is these things, they are all used, they are all used over biological systems, are used over biological systems in one way or the other, in one form or another, in one form or another. So that's advantage number two. Do we have another advantage? Yes, they also determine the specificity of a compound to be used. Very important. Point number three. Advantage number three is that they determine the they help to determine the specificity of a compound to be used. So they also help also help to determine the specificity of compounds to be used. Specificity of a compound to be used. Okay, so that's basically the the advantages of bioassay. The concept and the principles can be very useful when performing structure activity relationship studies as part of a drug intervention process. When you wish to compare the relative potency and functional effects of different compounds on entire tissue system. Testing of infected patient sputum help determine which antibiotic is given for quick recovery. It is a simple and faster method. Bioassay. It's a simple and faster method. The amount of test drugs available is small, does not involve complicated calculations, and it does not depend on dose response curve. Improvements in chemistry and biochemistry have increased the precision and accuracy of all physical methods for the measurement of the presence and concentration of drugs and other pharmacological active substances, have made many biological assays redundant. So bioassay, as like a fourth point you could say, is that it is what simple and it's a faster method. So we've seen the advantage. What is the disadvantage though? Does bioassay have any disadvantage at all? So we look at the disadvantage of bioassay in our next page. So right here we say the disadvantage of bioassay. So disadvantage of bioassay doesn't have any. Say the disadvantage basically is that the sensitivity of the tissue or sample you're working with could change with respect to time. The dosage timing may vary in mode of the application of the drug. It is less accurate, it is more time consuming and troublesome. This bioassay, the exact match of the response may not be available. Quantitative difference in between standard and test may not obtain and biological may not be obtained with biological variations. So let's quickly pen down some of these disadvantages. Number one, the biological sensitivity or sensitivity. Number one, the sensitivity of the tissue, sensitivity of the tissue changes with respective time, changes with respect to time. Okay. Number two of the disadvantage is that the dosage timing may vary. Dosage timing may vary in mode of application of the drug. Dosage timing may vary in the mode of application of the drugs, as when bioassay is used for pharmacological experiments or research. All right. Number three disadvantage is that it is less accurate. It is less accurate. It is more time consuming. more time consuming, less accurate, more time consuming, and troublesome. Number 4 of bioassay is that its exact match of the response may not be available. The exact match of the response may not be available. May not be available. So the quantitative difference between standard and test may not obtain any biological variations. So point number 5 of the disadvantages is that the quantitative difference between standard and test may not obtain any biological variation. So that's basically the disadvantage. We looked at advantages and disadvantages of what bioassay. We want to quickly see in these aspects of bioassay how they are applied. So in our next slide, we'll be talking about the applications of bioassay. The applications of bioassay. So applications of bioassay. One of the basic application is that it's establishing regulatory requirements for water quality. So with bioassay, you are able to establishing regulatory requirements for water quality. Water quality. So number 2, how is it applied? It could be applied in ecological monitoring of sex of sewage, ecological monitoring of sewage discharge. Point number 3 is that it could state environmental monitoring of water bodies, particularly in areas of the human exposure. So, state environmental could be applied in state environmental monitoring, of water bodies, particularly in the areas, particularly in the areas of the human exposure. Okay. Point number 4 is that it could be applied in environmental impact assessments of new technologies, treatments, facilities, reconstruction, and modernization of national economic projects. Okay, for designing local treatments, facility assessment of aquatic ecosystem. It could also be used for the determination of drug potency. Could be applied for the determination of drug potency, dosage, and side effects. Dosage, side effects, etc. Then you can also use it, like I said earlier, for environmental impacts, environmental impact assessments, of new technologies, of new technologies, um, treatment facilities, reconstruction, and modernization of national economic projects, and modernization of national economic projects. Let me write that properly. National economic projects. This is economy. National economy. Yeah, that's basically the applications. In our next set of lectures, we'll be looking at different areas in which biostatistics is important, the importance of biostatistics in biology, research, sciences, and medical science proper, when what area, what type of biological and medical data is important for biostatistics. We'll be looking at all of that in our next set of lectures. Thank you for listening.